Okay, good morning, everyone, or good afternoon, good evening, perhaps. Welcome, everyone, to day 2 of the NCI Symposium on Mutation Signatures in Cancer. And today we begin with introductory remarks by the Director of the National Cancer Institute, Dr. Ned Sharpless. Ned?
Good morning, everyone. It's a pleasure to be here today. I'd like to, kick off day 2 of a very exciting meeting on a very exciting topic. First off, I think we should thank the organizing committee who worked on this, putting these meetings together is a substantial amount of work, particularly Ron Johnson, Ludmil Alexandrov, and Terry Landy for all the work putting together a great presentation and program, set of presentations of the program. And I'd like to welcome all the speakers and participants. I think we appreciate your willingness to come and help the NCI think through this topic and provide scientific advice and talk about the cutting edge of research in this area. So thank you for your time and welcome today. I think it's a very interesting time in cancer research. It's, we've had this period of what I would call remarkable progress, where we've seen a nearly 30% decline in cancer mortality from its peak in 1990s. And that's been led by advances in treatment and prevention and screening and diagnosis. All of these things together conspiring to a really productive period. And it's been underpinned by a real basic science understanding of cancer that's much more sophisticated than we had, say years ago. And so this, investment in biological research is translating into accelerating progress for our patients. And I think we'll come to think of this period, right now, today, as a real golden age of cancer progress. And, on top of that, it's very galvanizing to have a president in the White House who has such a strong personal commitment to cancer and has stated that ending cancer as we know it is a top domestic priority for this administration. And so we're doing a lot of work at the NCI to think about what that means to end cancer as we know it and how the NCI can help lead that effort. And so within that framework, I think this topic of mutational signatures is a particularly important effort. It's a fascinating science, and I'm so interested in convening the meeting. But I think this work also has really important implications for lots of areas of cancer biology, and in particularly implications for the work of the National Cancer Institute and across the federal government. And so, the areas of mutational signatures are rapidly emerging areas with relevance to several parts of cancer biology, including understanding the etiology of cancer, understanding the role of exogenous exposures to cancer within certain populations. I think it has a very fundamental role in understanding cancer biology and helping us understand identify, for example, mutational processes arising during tumorigenesis. I think it helps us understand the effects of cancer treatments by monitoring, somatic changes within cancer genomes during therapy. And also, I think this developing field has pushed some really interesting computational develops in terms of developing new methods to detect and analyze mutational signatures and to study these aggregated data in complex ways. And I think that has been fascinating as well. So I think, this tremendously interesting science, though, has some important implications for what we do at the NCI. A good example of this is the area of environmental carcinogens. So this is a topic where the NCI has been, involved in the study of environmental agents that promote cancer for decades. But this is a very difficult field, I think, as this group is aware. It's very hard to ascribe causality to that, an environmental agent causes a cancer, and it rests upon this sort of time-honored troika of biological plausibility, rodent models, and then epi-cohort studies. And I think, this ability to ascribe specific signatures to specific mutagens is a very powerful additional technology that can help firm up some of those scientific linkages and address this ongoing public health concern. And this is an area that frankly is in some ways getting worse. We have a sprawling number of putative environmental agents that our citizens are exposed to. Lots of new data on these compounds. For the PFAS molecules alone, for example, there are thousands of individual different chemical species. So, it's a challenging scientific problem, and I think any additional tools that we can come up with to help understand that issue are very, very important. I spoke recently at an NIEHS meeting on this topic, and suffice it to say, there's a strong interest across many other parts of the federal government in making progress in this field, and a uniform position, I believe, is that somatic mutational signatures can contribute new links beyond, those traditional methods. And so I think that this is an area where we will continue to work with other parts of government, including FDA and EPA, in addition to other parts of the NIH. And we will look very much to this science to help drive that along. It is additionally an area that is of interest to the United Kingdom. So we had a recent a joint US-UK scientific summit at the request of Prime Minister Johnson and President Biden to facilitate cooperation between the two countries, and mutational signatures and the role in their detection in environmental carcinogen research was one of the topics of discussion. Some of the participants on this call were involved in that symposium as well. And I think this is one very good example of how this very interesting basic science can really inform the work of the NCI to promote benefits for the public health. And so stay tuned. I think we'll be doing a lot more in this area as time progresses to use this new science as well as other new scientific methods that are developing for better understanding of environmental carcinogen research. So that's why one of the reasons, one of several reasons why this meeting is so important. And therefore, I really am excited about the science, and I look forward to a great meeting. And again, I thank you for your time and participation in this so that we can all work toward the president's goal of ending cancer as we know it. Thank you.
Thank you, Dr. Sharpless, for those excellent comments. And now I think we're ready to begin the next session, and Dr. Jerry Lee will be facilitating that. So Jerry, go ahead.
Thank you, Ron. I'm Jerry Lee, a program officer at the Division of Cancer Biology of the National Cancer Institute. I'll be monitoring session 3. Exogenous and endogenous processes. We have 4 exciting talks in the session, followed by a panel discussion. Each talk is 20 minutes, followed by a 5-minute Q&A. Audience, if you have questions, please enter into Q&A. We'll try to answer as many as time allows. Okay. The first talk is given by Dr. Steve Rosen. Dr. Rosen is a professor of cancer and stem cell biology. He also directs the Center for Computational Biology at the Duke National University of Singapore. The title of his presentation is Two Signatures, one important for public health, one important for DNA damage and repair. Dr. Rosen.
Dr. Lee, thank you for the introduction. And I very much appreciate the chance to talk about our work at this symposium. So yes, today I'll be talking about 2 signatures, one of which is important for public health, and the other, I think, is important for our understanding of DNA damage and repair. So some context, we discover mutational signatures by mining mutation databases or by experimental delineation, for example, by treating cell cultures or mice with mutagens and then using DNA sequencing to identify the induced mutations. The signatures then can give us insights into molecular cancer epidemiology, and then from that, public health interventions and prevention or screening. Signatures can also give us hints about what mutational processes cause cancer and how cancers develop. And they can also expand our understanding of DNA damage and repair. So today I'll give one example from molecular epidemiology and public health, and one example that expanded our understanding of DNA damage and repair. So the public health example is something I've talked about before, aristolochic acid, we'll call it AA. AA occurs naturally in plants that are widely used as herbal medicine. So these are plants that are used in Chinese herbal medicine, but it's not just Chinese herbal medicine. This is the European plant, and in Romania, they claim it combats cancers, ulcers, prevents hair loss, and it's good for treating hemorrhoids. This is another species with the same AA in it that's made as a tea, used as a tea in Brazil, And this is another plant that's used in Indian herbal medicine. So in the 1990s, AA emerged as a human health problem when some 100 young women developed end-stage kidney disease. They had all gone to a single weight loss clinic in Europe. And in that weight loss clinic, they had switched from Hanfangji to Guangfangji. So two plants that are thought to be more or less interchangeable. Here's Hanfangji, it's dried slices of root. And here's Guangfangji, also dried slices of root. The first one didn't have any AA in it, and this one quite often has a lot of AA in it. Many of the AA kidney failure victims developed upper tract urothelial cancer. And by 2013, we and other groups had identified an AA mutational signature, both in upper tract urothelial cancers. So those are cancers in the tube that drains urine from the kidney down to the bladder. And we had found the signature in those cancers and in AA exposed cell lines. And I think we've probably seen a lot of signatures in the last day and today. But these gray mountains are the AA mutational signature, and these are adenine to thymine mutations in various trinucleotide contexts. By 2016, we knew at least anecdotally that there was AA exposure in liver, bile duct, kidney, bladder cancer, as well as upper tract urothelial cancer. So in 2016, we also knew that Taiwan was a hotspot for AA exposure and for AA upper tract urothelial cancer. So we thought it would be a good place to investigate AA in liver cancer in depth. And so we sequenced 98 hepatocellular carcinoma exomes, and we're pretty stunned to find that 78% of them had AA mutagenesis. So in this graph, these are exomes, remember, so this is quite a lot of mutations for an exome. So the height of the red bar here indicates the number of AA mutations in a given hepatocellular carcinoma. And here's the spectrum of one of these HCCs here, just for reference. So at this point, we've now been able to look at some 2,000 liver cancers, and we find that, in fact, yes, AA exposure is prevalent, especially in East Asia. And many AA exposed liver cancers have multiple known cancer driver genes with AA mutations. This is our data from 2017. So the y-axis is the number of AA mutations in known driver genes per tumor. And we see that Taiwan is really an outlier, but other countries and geographical areas also have substantial AA exposure. So there is now very strong evidence that AA can cause liver cancer. That evidence includes the fact that AA is a known mutagen and urinary tract carcinogen. There are multiple known cancer driver genes with AA mutations in liver cancers. Epidemiologists in Taiwan have shown a linear relationship between AA dose and risk of liver cancer in hepatitis B infected patients and in hepatitis C infected patients. And AA causes liver cancer in mice. And now we know of 6 cancer types with AA mutagenesis. So these include esophagus, liver, bile duct, kidney, upper tract urothelial cancer, and bladder cancer. This leaves us with some important questions. What is the dose risk relationship between AA exposure and liver cancer? And especially, is there an interaction with hepatitis? This would be analogous to the known interaction of aflatoxin with hepatitis in promoting liver cancer. How actually are people being exposed? Which herbs? How prepared? We really have very little idea. What is the extent of exposure in Africa and Latin America? We know from ethnopharmacology that these plants are used there, but we have no idea if they're causing cancers there or kidney failure there. What might be effective approaches to education and regulation? And how might we, in fact, convince some regulatory agencies that regulation is a good idea? Can non-invasive tests, for example, blood tests for AA exposure, be developed for clinical or research use? So, for example, for research use, right now, a big obstacle to understanding the dose-risk relationship is we don't really know how many people get exposed to AA but never get cancer. you only see A in the people who got cancer. And a non-invasive test would enable secondary prevention. Exposed individuals might benefit from screening. So to summarize, I think, put AA in context. We discovered the AA signature through mining cancer mutation databases and through studies in cell culture that established AA as the cause of the signature that we saw in upper tract urothelial carcinoma and then 5 other kinds of cancer. And finding this signature had eventually unexpectedly wide ranging implications as we found AA in those other cancers. And especially notable is the prevalence of AA in liver cancer, coupled with additional epidemiological and experimental evidence that AA, in fact, can cause liver cancer. So our second example involves a mutational signature that expands our understanding of DNA damage and repair. And this study came out of a very deliberate attempt to link mutational signatures detected in databases of tumors with experimental studies of DNA damage and repair in yeast. And this was studies of DNA damage and repair in the laboratory of Sue Jinx Robertson at Duke University. And specifically, we found a hotspot mutation in topoisomerase 2 alpha, which we'll call TOP2A, that causes a mutator phenotype in human cancer. So recall that topoisomerases cut and rejoin DNA strands to let them untangle or to let supercoiled duplexes unwind. And one mutant in the yeast topoisomerase 2 gene causes duplications of 4, 3, or 2 base pairs. For example, a 4 base pair duplication like this. So a GATA becomes a GATA, GATA, a CAT becomes a CAT, or a AT becomes an AT. And this turns out matched a signature that actually Ludmil had found in by data mining. This is a signature called ID17. ID17 is a indel mutational signature. And maybe people haven't seen as many of these plots before as they've seen the single base substitution plots. But this peak here, it represents the proportion of these 4 base pair duplications. This peak here, the lighter blue, represents the 3 base pair duplications. And this very faint blue peak represents the 2 base pair duplications. So ID17 was in 4 stomach cancers. These are their spectra. And then we found 3 other tumors with ID17. And this is then the average spectrum of tumors with ID17. All 7 tumors had identical mutations in a human ortholog of yeast TOP2, that's human topoisomerase 2 alpha. That was a lysine to asparagine at residue 743 in TOP2A. So we're going to call that K743N. So among some 12,000 tumors, only the 4 stomach cancers had 743N, and no other tumors had the ID17 duplication signature. So that's a very strong association between 743N and the associated signature, so P less than 10 to the minus 14. But besides the ID17 duplications, the 743N tumors also have duplications that are not in ID17. So these include deletions of 5 or more base pairs indicated by the height of this bar here. So this is the average spectrum of 743N tumors. So that might be something like a deletion of TACAG or deletion of CTACAGG. Okay, and these are deletions of 5 or more base pairs with microhomology, for example, CTA, and where the microhomology is the CTA here and here, or it might be a deletion of CTACAGG, where the microhomology is the C here and here. So these deletions rather resemble another signature called ID8, also previously discovered by data mining. So here is the average K743N spectrum. Here is ID8, and here's ID17. So ID17 doesn't have this big red bar, which are the deletions of 5 or more base pairs without microhomology, and ID17 doesn't have these purple peaks here, which are the deletions of 5 or more base pairs with microhomology. So ID8 is common. So we wondered, could these deletions in K743N tumors be caused by the same process that causes ID8, and these are just sort of co-occurring? Or conversely, are these deletions in fact due to the 743N mutation in TOP2A? So it turns out that K743N tumors and ID8 tumors have different distributions of deletion sizes. So here's an ID8 dominated tumor and the ID8 signature. And so you'll see that they don't have deletions of 2 base pairs, 3 base pairs, or 4 base pairs. So that's these peaks here, here, and here. They're in the 743N tumor, but they're not in ID8 or in an ID8 dominated tumor. And similarly, for short microhomology deletions of 2, 3, and 4 base pairs, that's these light purple peaks, they're in 743N, but they're not an ID8 dominated tumor or in the ID8 signature. So again, deletions of 2, 3, and 4 base pairs are much more common in 743N mutated tumors. 743N tumors and ID8 tumors also have different distributions of the sizes of larger deletions. So these red lines here show the proportions of large deletions according to their size. So this peak is actually deletions of 6 base pairs here. And then these other lines here that aren't red are tumors dominated by ID8 and another indel signature, ID6. So deletions of 6, 7, and 8 base pairs are much more common in 743N tumors than in other tumors. And conversely, deletions of more than 15 base pairs are rarer in the 743N tumors than the ID8 dominated or ID6 dominated tumors. So these differences in size distribution suggest that many or most of these deletions in the 743N tumors are actually caused by that mutation and not by the ID8 process. So here's the DNA damage and repair perspective. And this is from Sue Jinx Robertson's lab. So yeast and in vitro experiments show that the duplication signature in yeast TOP2 mutants occurs because the mutated TOP2 cannot religate after nicking. So these are the topoisomerases. They've nicked the DNA and that they've got these 2 strands separated with overhangs, but the mutated TOP2A can't put them back together again. And so there's various ways that the cell can rescue this. And we think that they all involve non-homologous end joining. So perhaps the cells fill in the overhangs and then ligate, or there could be some annealing followed by filling in the overhangs, or there could be some clipping back of the ends following by ligation. And these could produce 4 base pair, 3 base pair, and 2 base pair deletions, which is what we observe in yeast. Sorry, these are not deletions, these are insertions. This is what we observe in yeast. So this would create these duplications. So from the DNA damage and repair perspective, we think duplications due to human TOP2A 743N arise from the same processes as duplications due to the yeast TOP2 mutants. So that's these duplications of 4, 3, and 2 base pairs. But we think that the deletions of more than 2 base pairs, so that's deletions of 2, 3, 4, and more base pairs, and then deletions with microhomology. We think that, so we observed that human 743N tumors have deletions that yeast TOP2 mutants do not have. And so we hypothesize that the deletions in the human TOP2A 743N stem from repair by theta-mediated end joining. And that's the pathway that yeast do not have. We also found 2 sequence motifs associated with duplications and deletions in the 743N tumors. This is one group of motifs This is another group of motifs. These motifs here on the top of the slide, these are the 743N motifs. These motifs are not found at ID8 deletions in the ID8 dominated tumors. So this again is further evidence that many or most of these deletions in the 743N tumors are caused by that mutation and not by the process that causes ID8. We think that this K743N mutation contributes to oncogenesis. 6 of the tumors had presumably inactivating frameshift indels in P10 or P53 that were probably due to the 743N mutation. And also there was a known oncogenic deletion of BRAF in one of the tumors. One or both of the 743N sequence motifs were present at each of these sites. And in total, the 743N tumors had 45 indels in cosmic cancer driver genes, and one or both of the sequence motifs were present at 35 of the 45 indels. So the odds ratio for that enrichment is 177. To summarize, look at my notes here for a sec. To summarize, we've seen 2 examples of the causes and consequences of mutational signatures. And both examples involve data mining and experimental mutagenesis studies. And both examples provided new information on which mutational processes likely cause cancer. One of the examples, AA, is notable for its public health implications. And one of the examples, the top 2A K743N mutation, is notable for expanding our understanding of DNA damage and repair. So for the AA study in Taiwan, I'd like to especially thank Changgung Memorial Hospital and Xie Senyang, also Alvin Ng in my lab. And for the top 2A study, I'd like to thank especially Sue Jinx Roberts and Nicole Stantial in her lab, John Nittis in Chicago, and Arnaud Boat in my lab. And with that, I'd like to take questions.
Great, right on time, Steve. I see a couple of questions in the chat. Those are the questions from the panelists. The first question from Terry Landy. We saw AA signature in 15% of never smoker lung cancers from Taiwan. Could you speculate on how this plant-based exposure could generate a signature in the lung?
Okay, so just to clarify, Tara, you actually saw AA in lung cancers from Taiwan, is that correct?
Yes, I cannot start my video for some reason. It says that.
I don't know if I can see the answer somewhere. It's in the chat. Okay, sorry.
Yeah.
Okay.
I did.
So that's
did
amazing.
find-
So we can
yeah.
have, so we can say 7 cancers. Yeah.
Right.
Well, yeah. Okay, so a couple of things. We think that AA does get around in the body. I didn't talk about it, but we've done some work with TrinStrand that shows that we can see AA mutations in blood. So the AA might be circulating in the blood also. So of course the AA mutations we have to do with AA affecting hematopoietic stem cells, but it's, I mean, I'm just speculating, of course, Terry, but so one possibility is that it's actually getting around as a chemical in the blood or transported by some protein. And otherwise, I have no other actually speculation. And I just think it goes back to the, I would say that there are some signatures that mimic AA that might be in the lung, but probably not in other places. So,
Well, we analyzed over 1,200 lung cancer cases, and we found this signature very, very clear only in samples from Taiwan.
Taiwan.
Yes, only from Taiwan.
Anybody can do adductomics on that?
Maybe we could do it. That's just another idea we had is maybe they consume this herb like it dried up and maybe they inhale it, or
Oh, well, yes, there is evidence for that, actually. There is evidence. In fact, there have been some studies that people working with in herb shops, in traditional Chinese medicine shops, have elevated risk of kidney failure, presumably because of sort of exposure through dust from the herbs. So that actually is conceivable. I mean, we really, there's actually a lot of sociology that has to be done. I mean, I presume that even in the mainland, there's a lot of variation in how exposure happens. So we really desperately need some people in Taiwan, especially, I think, because it is such a hot spot, to look into these questions. Yeah.
Yeah, thank you.
Thanks.
Another question from Teresa in the chat. I would expect that mutations in TOP2A would cause replication stress and increase single-strand DNA, and that's a potential increase of AppleBack signature too. Is this observed as well?
Okay. No, well, in those human tumors with the K743N spectrum and the top and that particular mutation, we do not see AppleBack getting turned on. Well,
yeah.
I mean, it's actually there. I should double check that, but I'm pretty sure we don't see AppleBack turned on in those tumors. We do see. Yeah. Go ahead. Yeah.
Sorry.
No,
There's
okay.
a question in the Q&A from Sudhir. Most of the studies you presented are from Asia, exposure to some unique plants. How would you extrapolate this experience in the context of USA? I don't know if you have comments on that.
Well, so the exposure is overall rather less in the US as far as we've seen. We did see exposure in the TCGA data from patients treated at the Mayo Clinic, and we actually suspect that they came in from overseas, from Asia, for treatment at the Mayo Clinic, because otherwise exposure in the US is not very prevalent. So I think if you're the US taxpayer, maybe you don't want to pay for research into AA. So that's okay.
Thank you, Steve.
Thank you.
Guys, that we're running out of time for this presentation. We have to go to the next one. We cannot address our questions. I hope the speakers will be able to answer some of the questions after their presentation.
Yeah, will they be on? Will they be around? How will we find them afterward?
They're in the Q&A.
In the Q&A. Okay, thanks.
Yeah, if you go to Q&A and hit.
I got it. Yeah. Thanks so much.
Yeah.
Thank you, Dr. Lee.
Thank you. Okay, the next presentation is from Dr. Alan Bowman, who is a professor in cancer genetics at University of California, San Francisco. And the title of the presentation is The Impact of Carcinogens, Obesity, and Chronic Inflammatory Processes on Mutational Signatures and Cancer Risk in Mouse Tumor Models.
Okay, thank you, Jerry.
Yep, you're getting there.
Can you see the screen all right?
Yep, perfect.
Great. Okay, so this audience is particularly aware of the fact that the environment has a huge impact on cancer incidence. Using known mutagens, you've just heard from Steve about aristolochic acid. There's also a large number of suspected environmental carcinogens that have been tested over many years by the National Toxicology Program and others, chemicals in the plastics industry, pesticides, etc. In addition to that, there is radiation, a well-known cause of cancer after exposure to medical X-rays or radiotherapy. But there are other chemicals out there, for example, chemicals that may have tumor-promoting activity rather than being mutagens. We know very little about these, how many there are, where they are, and what their overall impact is. In addition to those, there are endogenous processes, inflammation, wounding, and so-called lifestyle factors, obesity, dietary factors, alcohol. And all of these factors together conspire to have a huge effect on human cancer incidence. So a long time ago, we decided that the only way to try to dissect out the contributions of these different components is to use a rodent model where we can treat animals separately with or in combination with these different factors and look at the effect on cancer incidence and of course now on mutational signatures. So what I'd like to do is go over, first of all, some work done a number of years ago on radiation. Most of the work on the mutational signatures was done by a very talented MD PhD in the lab, Rose Lee, who's now just taking up a new faculty position at City of Hope. So the radiation work, just quickly run through this. This has been published so you can check the details. We noted a long time ago that a germline deficiency in P53 can predispose mice to tumor development after a single dose of radiation. And this is a study using two different flavors of radiation, gamma or high energy radiation with ions. And both of these cause tumors in mice after a single exposure. The gamma is very good at causing early onset lymphomas. The heavy ion beams give early onset mammary cancers, and both types of radiation can cause lung and skin cancer. So these are very potent carcinogens. It's one single dose in the context of germline deficiency. If we look at the mutational signatures and mutation burden in these tumors, the number of point mutations is extremely low. It's right down at the low end of the spectrum in comparison with many of the tumors from TCGA. So we don't get many point mutations. However, the radiation tumors have a lot going on in their genomes, as you can see from this CNV slide. There are many large indels, aneuploidy, chromosome duplications, chromothripsis. And what you see here is a bunch of tumors, some lymphomas, mammary tumors, or the main bulk, and skin tumors. And just to home in on one example, in the mammary tumors, we see on chromosome 6 this high focal amplification on proximal chromosome 6. This is very distinct, and mainly, as you can see possibly from this slide, most of the tumors with the amplifications are caused by heavy ions rather than gamma. There are some induced by gamma, but most of them are due to heavy ion exposure. Turns out that this is a complex set of rearrangements that lead to progressive amplification of proximal 6, and the critical gene there seems to be the MET gene, MET oncogene, which is hugely overexpressed, as you see on the right, in tumors that have these copy number gains. So the conclusion from the radiation experiments is that there are very few point mutations, so no real point mutational signatures of radiation. But there are focal structural variants, chromothripsis, and these are the real hallmarks of radiation-induced malignancies. A second study that we did was in collaboration on suspected, known or suspected human carcinogens. This was with the National Toxicology Program, Robert Sills and Arun Pandiri and Ron Herbert. And what they've done is look through, I think, over 600 chemicals over the last 40 years. And they've done these very expensive and time-consuming 2-year chronic inhalation, chronic gavage feeding to mice. Harvested the tumors, and we obtained a large number of these tumors for sequencing at the Sanger Center. And just very briefly, this is the complex panel here. But what you can see is in the top panel here is mutation burden, and the high bars here is a subset of tumors that have a very high mutational load. This is up to 20,000 mutations here. And we see these in the lung, the liver, the kidney, some forestomach tumors here too. These are the mutational signatures, which do show some tissue-specific preferences for mutational signatures. But the interesting thing is that most of the tumors did not have very many mutations. And in fact, the arrows heads here show spontaneous mutations in the same tissues. And most tumors induced by these chemicals were indistinguishable from spontaneous tumors. And this was published last year, excuse me, in Nature Genetics. And this caused us to focus on the possibility that many environmental carcinogens may in fact be acting as tumor promoters as mutagens. And this is a slide I stole from Paul Brennan, who was a co-author of this paper by Sarah Moody from the Sanger group, showing that esophageal cancer, this was mentioned yesterday too by someone, esophageal cancers from very high risk and low risk areas across the planet did not have any difference in their mutational signatures. So no obvious smoking gun of a carcinogen that's causing this difference in high risk. So these two papers together from Mutagraphs, I think, are suggesting that non-mutational processes also play a very important role in cancer. That may be heresy for some of the people in the audience, but this has been information that's been out there for many decades. And if I can just briefly show this slide, a summary of work done in the 1940s by Isaac Bernblum and Phil Shubik using a skin model, where they showed that exposure of mice to a potent carcinogen by itself does nothing for up to a year. Exposure to a promoter does nothing, but sequential exposure, excuse me, frog in my throat this morning. Sequential exposure to the mutagen followed by the promoter gives lots of benign and then subsequently malignant tumors. The critical experiment that Isaac Bernblum and Phil Shubik did was to treat with the mutagen and then wait for various periods of time, up to 18 months, most of the mouse's lifespan. It turns out that the initiated cells caused back here are still there after a year and a half, and they can still be promoted after a year and a half and give rise to tumors that will kill the animals if they don't die of old age before that. So the rate limiting step in this model is promotion rather than the presence of the mutation. So we've now gone back and sequenced a number of tumor models based on these kinds of results. This is the standard initiation promotion model up here. This is the Phil Shubik and Bernblum delayed promotion model. A different model involved from Klaus Gartler's lab involved embryonic exposure to the carcinogen, followed by promoter treatment of the adult pups once they've grown up. The effects of obesity and a high-fat diet, chemotherapy, which I won't have any time to talk about, and finally, tumors induced by genetic activation of RAS genes, followed by TPA promotion or skin wounding. So I'm very quickly, obviously, going to run through the highlights of some of these and try to give you the take-home message. The mutational signatures from these skin models initiated with DMBA are shown here. You'll recognize the patterns rather than the writing here, which is very small. But a prominent feature here is this signature here of A to T mutations. This is the signature caused by DMBA, which binds to adenosine residues, and codon 61 of RAS is CAA, so this gives us CTA. This is a mutation found several decades ago. And the signature here is very similar to 22, which is aristolochic acid, or 25. And Ludmil, who helped substantially with the analysis of this, used a pentanucleotide signature, to distinguish the DMBA mutational signature, for example, from aristolochic acid. But they are remarkably similar, suggesting that there may be polycyclic hydrocarbons out there that are causing signatures similar to aristolochic acid. One feature from this model, which was not obvious from other models, is that when Rose was doing the analysis of the total number of DMBA mutations, which goes up to as many as 50,000, and other mutational signatures, she found this very strong correlation between the DMBA signature mutations and SBS5 mutations. She did not see this for, this is one of the clock signatures, she didn't see it for SBS40 or SBS1. And it could be, we think that, and I'd be interested in other people's thoughts on this, that the chronic exposure, I can't see this myself here, move this around a bit. The chronic exposure to a mutagen, it's very difficult to distinguish the DNA damage caused by chronic exposure from time, because it's constant. This model that we've used involves a single dose of the mutagen followed by a non-mutating promoting factor later. And what she's seeing is that SBS5 is proportional to the number of mutations caused by this dose of DMBA, suggesting that there may be some kind of genome shattering effect, maybe activation of fragile sites that leads to SBS5, but we have absolutely no proof of that whatsoever. The various other models, this is the standard DMBA TPA model, the blue bars here, purple blue bars. This is the signature for DMBA. There are various other signatures here of reactive oxygen and the clock signatures. The important point here is that the delay model, this is after at least 6 months, some of these animals are 9 months out, all of the DMBA-induced mutations are still there, and they're still promotable by the TPA. The mutation signatures, the mutation burdens are in fact indistinguishable from the standard model. One difference was seen with this model over here. This is the embryonic treatment where mice are treated at day 12, 14, 16 during embryonic development. These have fewer DMBA mutations, and we think this is because it's a gavage treatment, and the absolute dose of DMBA that gets to the skin of the pups is low. But nevertheless, these hang on, something's happening on my screen here. These tumors induced by embryonic exposure to the carcinogen did have a higher incidence of reactive oxygen, suggesting that when cells are proliferating, as they are during development, they may be prone to DNA damage, which involves generation of ROS. Finally, obesity and alcohol, and then the final model will be the inflammation and wounding. Again, we can make obese mice by crossing fat mice and thin mice, different strains and species. And the take-home message here is that there is no obvious mutational signature of obesity. There is, however, a signature of inflammation, both in the normal tissue from these animals and in the tumors from the animals. The low body mass index or low fat diet mice, particularly the low body mass index mice, have a signature that suggests increased immune surveillance, increased involvement of the immune system, particularly in the normal skin, but also in the tumors from these mice. The high body mass index mice have more signatures of metabolism, protein synthesis, and so on. And finally, the role of tumor promoting factors and inflammation and wounding. Now, this is a model, this is the minimalist model. We can induce or initiate tumors genetically in a stem cell population. This is the skin. These are LGR6 positive stem cells in the isthmus region. This is the sebaceous gland. We can switch on KRAS, a very potent oncogene in these cells, and we get nothing. But if we treat with TPA, we get papillomas. If we do a single full thickness skin incision wound and then clip the wound at 3 sites, we get these huge papillomas that grow at the wound sites. These will rapidly progress and kill these animals. When we sequence these tumors, these are the DMBA, these are the embryonic, just turned on its side here. These are all the DMBA tumors. These minimalist tumors are shown here, and they have almost no point mutations apart from the initiating driver mutation. But I think you saw from that last slide how dangerous these tumors are, although they have very, very vanishingly few point mutations. They do have a number of signatures of endogenous processes, but no mutational signature attributable directly to wounding or to TPA. So this leads us to the conclusion that the rate limiting step, as mentioned 80 years ago by Bernblum, is the promotion step rather than the mutation step. And just finally, I don't know how, I can't see my time here, but I'll carry on until somebody shouts at me. This is the model that we're pursuing. So if
5 minutes left.
Sorry?
5 minutes.
5 minutes, excellent. So the model is that spontaneous or carcinogen-induced mutations will happen in normal tissues, and this can induce a range of different driver mutations. If promoters aren't causing these mutations, we think they are selecting from among a panoply of pre-existing mutations. For example, if this tissue is treated with promoter number 1, then it will clonally select this particular driver. If it's treated with promoter number 2, then a different driver is selected in a different tumor model. comes out. And there's reason to believe that this is the case from the skin classical studies on skin, showing that different promoters of skin tumors have a propensity to induce tumors that carry different driver mutations. Many carry HRAS mutations, but some have KRAS mutations. And just to highlight this mutation selection model, this is the study from the Riva paper in Nature Genetics. This was not highlighted sufficiently in this paper, but what this involved was sequencing of spontaneous tumors. And these are the driver mutations along the bottom here. You can see these different driver mutations, HRAS G13R, for example. This one here, that's the most common mutation, the biggest circle in spontaneous liver tumors. In, where is this now? Why is this not moving on? Ah, okay. Most of the carcinogens that gave rise to liver tumors did not induce the BRAF mutation, the HRAS G13 mutation, but they induced HRAS Q61 mutations. In the lung, similar picture, the most common mutation in spontaneous lung tumors is BRAF, but many carcinogens induced FGFR2 mutations, and cobalt out here induced preferentially a KRAS Q61H mutations. So although these pre-existing mutations are spontaneously occurring, they're not caused by the carcinogen. The carcinogen is selecting from among these pre-existing mutations. So this is exactly the model here, whereby in the skin, as I mentioned earlier, TPA induces tumors with HRAS mutations. Other promoters induce tumors with KRAS mutations. And just the very last slide, we can now begin to demonstrate these features using short-term in vivo assays. This was with TwinStrand, and this is TwinStrand sequencing of a normal mouse. These are the mutational signatures, very little there. A single exposure to DMBA gives the classic DMBA mutational signature I showed you earlier. This is 1 to 2 weeks after a single treatment with DMBA. That whole signature persists for a year. A normal mouse after a year has some additional mutations, and these are SBS1 related mutations. The TPA, these are two different animals, different mutations we find after DMBA, and the TPA is selecting this large pink circle, this is clonal selection, and that is in fact the codon 61 mutation that we find in most tumors. So we do believe that the role of the promoters in this is to select from among a pre-existing set of spontaneous or induced cancer-driving mutations. So I'd just like to finish here. Rose Lee did the mutational signature analysis, and Eve Kandaba has been doing the TwinStrand work and most other things in the lab, actually. And this has been a big team effort with many people in the group, and it's funded by Mutagraphs, the CRUK, as well as the NCI. Thank you.
Thank you, Alan. That's great. Right on time. I see a couple of comments, questions in the Q&A. The first is a comment which says pathogenic germline P53, P10, RB variants cause early onset cancers with very high penetrance. So promotion is clearly not a limiting factor. Do you have any comments on that?
Well, absolutely. I mean, the first few slides I showed that, because if you have a germline defect in P53, which can be heterozygous, or it can be a delta proline mutation, then a single dose of a carcinogen can, on its own, give rise to tumors in these animals, including some skin tumors. So clearly, powerful germline defects cause huge genomic instability, which seems to be much less dependent on the activity of promoters. And promotion is also a tissue-dependent phenomenon. I think the importance of promotion varies between different tissues, as we can see from some of the mouse models.
Okay, great. Next is a question regarding P53 model. Since P53 deficiency leads to aneuploidy and MET amplification, does that suggest that attempts to repair p53 with small molecules in cancer patients will not be successful?
Well, my understanding is the attempts to repair p53, which I believe so far have been unsuccessful anyway, the idea there is to reactivate the function of p53, which would induce apoptosis and kill the cells, regardless of the genomic status of the cells. That's the concept, I believe. And that may still work, regardless of the effect of loss of p53 on genomic instability, if they still retain that ability to be killed by a functional p53, if I understand the question correctly.
I also have a question about the mouse models. If different mouse strains treated with the same condition, do you observe a significant difference in terms of the signatures?
This is something we have looked at. And in fact, the animals, the DMDA signatures I showed were from a heterogeneous mouse population. And there we could not see anything obvious in terms of differences in signatures according to genetic status. But we still need to do a lot more on that particular question. I think it was almost certainly the case that genetic background affects mutational signatures. We know that it affects the selection of different cancer driver mutations, because on a B6 background or a spretis background, then even with the skin model, and it's also seen with the lung model, if you use the same carcinogen, the proportions of the different driver mutations can vary according to the genetic background of the host. So these things are all kind of interacting in complex ways, and it's an area that really needs much deeper study.
Yes, indeed. There's also a question in the chat. Oh, this is a rather long question from Terry Landy. In lung cancer from smokers, we saw a significant increasing of SBS4, DBS2, and ID3, all tobacco smoking signatures, with increasing pack years of tobacco smoking and decreasing with years with quitting smoking. These results highly correspond to epidemiological observation. Don't these results suggest that tobacco smoking involves more initiation than promotion effects in lung?
I don't know. I don't think we can conclude that in a quantifiable way. There is evidence, for example, that, well, clearly smoking is linked to lung inflammatory disease. Inflammatory diseases have promoting activity. Nicotine itself, I believe, there's literature showing that nicotine is immunosuppressive. So I think smoking may be such a great carcinogen because people are doing the initiation promotion experiment on themselves all the time. Clearly it's mutagenic, but in addition, you're putting something into your lungs that normally shouldn't be there. It's causing toxicity. It's causing some kind of a physiological response, which is likely, I think, to be promoting, but it has not been quantified.
Okay, thank you, Alan. Great presentation and great answers. We have to move on to the next presentation. So the next speaker is Dr. Serena Nick Zainov, who is a clinician scientist at MRC Cancer Unit of the University of Cambridge. The title of the presentation is Mutational Insights from Systematic Studies in Human Stem Cell Model Systems.
Thank you very much. I hope you can see my slides and you can hear me. So thank you to the conference organizers.
Perfect.
Okay, perfect. So thanks to Ludmil, Terry, and Ron Johnson and the NCI for the opportunity to present today. I understood my brief was to talk about some of the experimental work that we've done. So today I'd like to share with you some of the high-level insights that my team and I have learned from exploring endogenous and exogenous mutational signatures in 3 studies of human cellular model systems and in 1 clinical case. I won't be able to cover details in 20 minutes, but I will signpost where you can get them along the way. So let's do the first two studies to start off with. So we teamed up with David Phillips and Jill Cucab in King's College London to investigate the patterns associated with exposure to IR class 1 and 2 known or suspected environmental mutagens. We used human IPS models, and they were treated at a dose that achieved 40 to 60% viability. Cells were allowed to recover and then re-expanded over 7 days. Remember, each cell will have its own mutational accumulation. So to see those mutations that have arisen in any cell, we needed to grow them up from single cell subclones, and we aimed for 2 to 4 subclones for each treatment. And these were whole genome sequenced. Functional DNA damage assays were also performed, and in all, we had 13 families of compounds, 125 treatments in total of 79 agents, some with, some without metabolic activation at variable concentrations too. We used 15 different controls because the different solvents are carried in, the different mutagens are carried in different solvents. And so this, there was a large total of 324 daughter subclones that were whole genome sequenced. But because this was all done in an isogenic human cellular model, the data is all comparable across the system. So, in gray are the controls down here. And when you just grow cells in culture, you're going to accumulate some mutations. And the question was whether the treatments would produce profound patterns, which they did. We had clear patterns of single substitutions, double substitutions, as well as indel patterns. And in all, we had 41 substitution signatures, 6 double substitution, and 8 indel signatures altogether. And as I said previously, I won't have enough time to sort of show you all of them, and that would be frightfully dull anyway. All the data that was produced by this experiment is widely available through this website. You can browse the site interactively and look at things down to individual subclones. So warts and all are all on show, and you can download the data. And I was very pleased to hear that other people are now sort of starting to show this data in other websites as well, which is really super news. But as I mentioned previously, I really wanted to communicate some of the high level principles. So the first thing is I can say that the positive controls, those signatures associated with UV and aristolochic acid that you've heard so much about today already, those were faithfully reproduced. So here's the cancer signatures, and here are the experimental signatures. They're really very clear. The second thing to say is that can be some really humbling surprises. And we heard in Professor Beaumain's presentation previously about how there are some signatures that look a little bit like aristolochic acid. So the signature above here is aristolochic acid, and down here, this is DBPDE, which is a compound from the family of polycyclic aromatic hydrocarbons. That's tobacco smoke, and it had a similar pattern, which we initially dismissed as an error. We repeated the experiments at different concentrations, including a sort of an earlier version of DBPDE, the sort of DBPDE is a dialyl-epoxide, so DBP was used. And as you can see here, they really produced a very similar pattern to aristolochic acid. And if we didn't do the experiment, we wouldn't have known. So it's been quite an insight. And of course, if you looked at signature 4 from lung cancers, from Ludmil's work, for example, you can see the little peak from DBPDE contributing to that signature. The third thing to say is that the effect sizes of different environmental agents of the signatures is very variable. So prior to any of the mutational signatures work in 2012, 2013, there was already a lot of work out there on smoking or UV aristolochic acid. We knew a lot about them. We could see them in a dish. We could see them in cancer, and that's because the signal is very large from these agents. So some things can produce a very large effect size, and so they're easily detectable. Whereas there are other, including environmental mutagens, where the effect size is much smaller and perhaps not as easily detectable. Just coming back to the point made by Professor Rosen about environmental, about using this in a public health way, that actually the detectability is different from one environmental agent to another. The fourth thing to say is that some of the environmental agents can produce not one, but multiple mutational signatures. And these are the mutagens that produce substitution as well as indel or double sub patterns as well. And the implication there is that the environmental agent may produce a primary DNA damage, a form of a DNA adapt, but the final mutagenic outcome is a function of the various replicative and repair pathways that come into operation to fix that damage. And so you get multiple different outcomes. So as an example, we can dissect the signatures associated with 1,2-DMH. This is an alkylating agent. And I think this example also highlights how deep analysis can reveal insights into mutational mechanisms. So 1,2-DMH treatment results in a C to T substitution signature and a C indel signature. And the deep analysis revealed that this occurs at strings of T. So you have TTTT and they get a C. So that is very similar to a G at the end of a string of A's. And alkylating damage from 1,2-DMH would result in an O6-methyl G highlighted here in the red asterisk. So O6-methyl G would tend to pair with a T instead of a C, and in the next round of replication would result in this sort of a substitution. An alternative route to repair could also involve slippage of that polynucleotide repeat tract. A small IDL is formed. The O6-methyl G will still pair with a T instead of a C, but the net result after replication looks like a G deletion at the end of the poly A tract. So although there was one form of damage, there were two routes in which it was repaired, resulting in these two different outcomes. And this example sort of reinforces that fifth point. We aren't just interested in finding patterns, we're kind of quite interested in how those mutations actually arise. And continuing that theme on deriving mutational mechanistic insights, this group of individuals, we performed a separate systematic study of signatures of knockouts of all of these DNA repair and replication genes. Now, in this study, all we did was knock out the gene. We did not add any form of DNA damaging agent. So there's nothing to change what is a fundamental signature. And so if all you do is knock out a gene and it produces a pattern, then those genes must be really critical guardians of the genome. And again, I don't have time to go through all the signatures, but I can say that these are some examples of some of the signatures from genes involved in the basic excision repair pathway, and on the right-hand side, the mismatch repair pathway. So let's stay with mismatch repair. Again, high-level message. I think we can go beyond just thinking about mismatch repair deficiency as microsatellite instability or high TMB. There is a higher granularity of insights that we can distill, and we can even see some gene specificities. So here, knockouts of MSH2, MSH6, and MLH1 qualitatively produce a very similar pattern, and it's very different to the PMS2 knockout. Now, still staying with that, although they have a qualitative similarity, quantitatively, MSH2, MLH1, and MSH6 behave quite differently. So I draw your attention to the ratio of substitutions to indels of MSH6, really quite different to MSH2 and MLH1. And through the analyses of genome dynamics, replicative and transcriptional strand biases, +2, -2 sequence context, as well as neighboring motifs, longer neighboring motifs, we can work out the root cause of the diverse components of this signature. So in this study, we worked out that the C2A component was likely to be caused by guanine oxidation. The C2T and T2C components were likely due to replicative polymerase alpha, delta, and epsilon misincorporation. And this little tiny T2A peak here, modest, but a very definite peak, and seen in all of the experimental knockouts without fail. This is likely due to a new mechanism which we propose called reverse template slip, or due to double slip. And I shall leave you to fish out the mechanistic propositions from the paper, which I have grayed out on the slide. Knock yourselves out there. But just to recap on some of those messages, here we have used isogenic human lines. The results are entirely comparable to one another. Known signatures are recapitulated. New humbling signatures and insights are revealed. There's variable effect sizes of diverse signatures. And I think that's a little bit more nuanced, right? One etiology can produce many signatures, I showed you earlier, and one signature could have multiple potential causes. I think we get insights into mutational mechanisms, and all the data is available for browsing and download. So I think the bottom line is characterize your cellular models. For scientists who are studying the signatures, endogenous or exogenous sources, new drivers can arise in your cells in culture for long periods. And for the clinical scientists, all we do is karyotypic pre-screening, so copy number pre-screening before we use these cells for clinical trials. And really, this paper sort of urges caution on that note. And so in my last 2.5 minutes, I'm going to finish off with a clinical case where exogenous and endogenous mutagenesis comes into play in the interpretation. So this is Joshua Barnfather. I have his consent to tell his story. He's actually a patient known to me due to the DNA repair clinic. So he's got xeroderma pigmentosum, and these kids can't fix UV damage. They die from skin cancers in the past, but these days they can look after their skin, so they live into their adulthood. And poor Josh presented with a violaceous tumor in the middle aspect of his left eyebrow. It was an angiosarcoma. Now, he didn't seem to respond to any of the usual treatments for angiosarcoma. And by July 2018, this man was dying. So when we do his whole genome, we find a huge amount of UV mutagenesis. His drivers are pretty boring, CDKN2A and TP53, but his mutational profile looks like a malignant melanoma. It's a huge amount of UV mutagenesis, exogenous mutagenesis, but that's not what's killing him. And this is why the interpretation is important. He's an XP mutant, and so he cannot fix UV damage. This UV damage is normal for him. What is killing him is this signature 10, caused by polymerase epsilon proofreading mutations. We struggled to find it, but we did find the cause caused by UV. So that's the Paul E driver mutation causing this. It was in a subclone, however. Now, we then did PD-L1 staining because polymerase epsilon mutated cancers are supposed to be sensitive to checkpoint inhibitors. We did PD-L1 staining in the primary tumor, but that turned out to be negative. But remember, I said that this might be in a subclone, so perhaps this is a false negative. And what's killing him are the Mets, it's not the primary. So we then repeated PD-L1 staining, but this time in the Mets, and this lit up really strongly. So there was huge PD-L1 positivity. He got his pembrolizumab. So these were his scans pre-treatment. This man was really dying at this stage. But with pembrolizumab, after 3 cycles, things were just melting away. After 7 cycles, everything melted away and this man walked home. So this is an anecdotal story, but really the bottom line is that when we think about endogenous and exogenous mutagenesis, we need to think about it in the context of the whole patient when we're interpreting it, including the germline information. So that was me. I sort of galloped through an account of our experimental studies in human cellular models. I talked about the IPS stuff with the BCOR variants, and then I finished off with a clinical case. I'd like to thank an extensive list of collaborators at the bottom left-hand corner, and of course my generous funders, my highly international and very diverse team. We even have an elf from whom I draw all my inspiration. I haven't seen some of them for a long time, and I'd like to re-acknowledge their grit, resilience, and determination through this really interesting time. Thank you very much. I'm happy to take any questions.
Thank you, Serena, for keeping it on time. There are a number of questions both in the chat and in the Q&A. We try to go through several of them. In the Q&A, there's a question here from anonymous attendee. What do you think are the underlying reasons for the discordance between the experimentally produced aflatoxin mutations in cell lines, in QCAP, etc., and SBS24 in the catalog? Does this mean SBS24 is not caused by aflatoxin directly, but as a result of an indirect metabolic product?
Okay, great. Thank you so much. So there's a couple of questions in there as to why the discordance in the first place. I don't know exactly what is the reason for the discordance. I would say that if you use different cellular models, you might get slightly different outcomes. We know, for example, that platinum in DT40 is very different to platinum in humans. So, I think your cellular model probably does make an impact. So could it be that it is more of an indirect pattern, and perhaps in our cellular model, we don't have the appropriate metabolic activation to permit the aflatoxin signature to show itself? That is perfectly possible. So I think with this and with any other study, right, I think we really do need to think about what models we use. These are human stem cell models as opposed to cancer cell line models. So I think if you did the same experiment in a cancer cell line, you might see a slightly different outcome.
Okay, next one. How long UV damage persists in iPS cells?
I'm not sure I fully follow the question, but I'll try to answer it anyway. In our study, it was a really short burst of UV damage. We just give a burst of UV damage, let the cells recover. The pattern, the signature, will persist as long as it's been imprinted in the genome. So that's not going to go away. So you would see the pattern of mutagenesis pretty much indefinitely. If you give further bouts of UV damage, you might see more additions of an increase in the mutational load associated with the UV damage. I'm not sure I answered that question, but I tried.
Okay, thank you. There's also a question in the chat. This one's from Teresa. to what extent similarities between signatures of environmental exposures might be related to common DNA repair pathways?
This is a great question, actually, a huge one to answer. I didn't have time to go into it today. And some people have said that the signatures that you see from environmental exposures, because they're so huge, might actually be due to DNA repair pathways transiently sometimes not work so well, and that's why you get a huge amount of mutagenesis. Because our cells are supposed to not allow so much mutagenesis, right? That's why we have so many DNA repair pathways. I do question that thinking slightly, and I'll tell you why. It's because we know that the DNA repair pathways must be operative. For example, when you do the experiments with UV or smoking, and we see it in cancers, you can see the activity of transcription coupled repair. So TCR is working even in the experimental models. So that's intact. Now, if you had mismatch repair deficiency and HR even transiently, you will see the signatures. They are huge. So again, you would see those patterns, and we don't see them. So I would guess actually that the fact that we can see such marked mutagenesis from environmental agents is simply due to the amount of damage that you're getting. So if the dose that you're getting is large, or the effect size is big, then you will see a big and clear pattern. So I think actually the DNA repair pathways are doing exactly what they're supposed to do. They just can't fix all the damage because there's just so much of it coming.
Okay, thanks. Again, we're running out of time. Thank you for your great presentation. And there are a couple more questions in the Q&A if you can get your answers in there.
Sure, no problem.
Okay, thank you. All right, now we're getting to the fourth presentation from Dr. Ruben Harris, who is a HHMI investigator, a professor of biochemistry, molecular biology, and biophysics at the Center for Mutation Research of the University of Minnesota. And the title of the presentation is Insights from Inflicting Pure APOBAC3 Mutation Signatures in Human Cells.
Thank you very much. I want to commend the organizers and the other speakers for an amazing meeting so far. This is a picture of fall at Minnesota. Very soon, this will be blanketed in beautiful white snow. So my lab studies APOBEC enzymes and APOBEC-mediated mutagenesis. APOBEC enzymes, we often use this word in the singular. They catalyze the deamination of cytosines to uracils in single-stranded DNA, which is a relatively simple water-driven chemical reaction. Although the mutagenic outcomes can be quite diverse, as I'll explain, and you've seen in some of the other talks already. I want to remind you that the word APOBEC comes from apolipoprotein B mRNA editing catalytic subunit 1, which is a mouthful, but it is the founding member of the family, and that does have an RNA editing activity, and many of the others are DNA specific with really unique biological functions. So we're interested in APOBECs and their antiviral functions. That won't be a subject of today's talk, but it's important to note why this family is growing and amplified in some species, including humans or higher primates. And viruses, of course, encode, or successful viruses encode ways of fighting back. And that's a major topic in my lab. And over the years, we and others have found that the APOBECs are contributing largely to cancer mutagenesis as well. And I wanted to do a shout out also for APOBECs in genome engineering. So tethered to Cas9, they can be used to selectively edit cytosines in any DNA-based genome. And that will be a topic of an upcoming meeting. So if you are in a winter climate like I am, next month in January sounds quite appealing, and the meeting will be in person. And so today's talk, and if you just Google deaminate, you'll find it. Today's talk will be focused then on two areas. So human APOBEC3s in mice and human APOBEC3s in human cells. And the larger question is, which APOBECs are involved in cancer mutagenesis? And so in humans, we have actually 9 active single-stranded DNA deaminases, AID that's important for antibody diversification, APOBEC1 that I mentioned, that edits the ApoB mRNA, and then the APOBEC3s that have overlapping roles in innate antiviral immunity. And so shown down here is actually an expansion of a co-crystal structure of APOBEC3 B in magenta and A in cyan with single-stranded DNA, so that you can see how this single-stranded DNA is bound by the enzymes with the target cytosine deeply in the active site pocket, the -1 thymine position between these 2 loops. And there's actually 3 hydrogen bonds that are important for this, and that explains the selectivity of these 2 enzymes for TC dinucleotide sequences. So we know, and we've heard a lot already, so I won't belabor this, that APOBEC is promoting many different cancer types, or at least associated with many different cancer types. So these are data for APOBEC3B, where we're looking at a heat map, and overexpression is shown in orangish reddish color relative to normal levels in a cool blue color. And one can see that APOBEC3B is overexpressed in many different tumor types. We've seen the signature displayed many different ways, and this is just looking at the trinucleotide context of APOBEC mutations accumulating preferentially in TCA and TCT motifs. And the mutational outcomes are C to T transitions, predominantly in C to G transversion mutations, depending on how those initial uracil lesions are resolved. And we infer that it's clinically relevant because in multiple different tumor types, this is estrogen receptor positive recurrent breast cancer. High levels of APOBEC3B associate with poor clinical outcomes, and lower levels associate with better clinical outcomes. This is just an analysis of 2 larger data sets, actually. So Serena's talk, that you all just heard, it was beautiful. And these are also data from one of her studies, where we're looking at 560 primary breast tumors. And one can see the different signatures shown here, including the HRD signature and the APOBEC signature. And the main point here is there's a lot of mutations, often inflicted by APOBEC. There's the C to T and C to G in 2 different shades of pink. But the other point is in the metastatic setting, the APOBEC signature is actually a much larger component of the overall constellation of single base substitution mutations, as is the HRD signature, and that these are largely mutually exclusive. So a working model that we've been really developing and testing over the years is that normal cells don't have high levels of these mutagenic APOBECs. Virus infection, inflammation, cell cycle dysregulation can all lead to higher levels, which would then promote increased mutagenesis and increased tumor evolution. So one of the big questions, seeing all of these APOBEC mutations all over many different cancer genomes, is it a passenger or a driver? And yesterday we heard too that in many different tumor types, APOBEC signature appears later in tumor development as opposed to earlier. And so this is a major question. And we've aimed to address it a couple of different ways. And one is shown here where we're using the well-known FAH model for liver tumor development, where one can essentially, through hydrodynamic injection, deliver a DNA construct to the liver that's then taken up by virtue of selection for FAH positivity. And then over time, one can look at the impact of payloads, which can be knockdown of tumor suppressors or other things like APOBECs delivered in that same construct. And so when we did this experiment by injecting it 2 months post-birth, and following this for 6 subsequent months, the data were really quite striking. So these are livers pulled from animals that were injected with constructs expressing either empty control or the different APOBEC family members shown here. And one can see high levels of tumor formation from APOBEC3A, but not any of the other APOBECs above that background. What's additionally notable here is that A3A is sufficient. So in the initial experiments, we did this in combination with P53 knockdown. That's not necessary. Catalytic activity is required. It inflicts a strong signature that I'll show you shortly. These other APOBECs are not carcinogenic in this model. And really importantly, we were initially puzzled by APOBEC3B being as negative as the other controls in this experiment. But further incubation of essentially this model in cages allowed the accumulation of APOBEC3B driven tumors. So a little bit longer, and that's consistent with biochemical data and structural data showing that this enzyme is a little bit less catalytically active, 5-fold or so. And so in parallel to that, we were interested in developing other models for APOBEC-driven mutagenesis in mice. And 2 of them are depicted here, where we've knocked in an APOBEC3B minigene downstream of a transcription stop cassette at the non-essential ROSA 26 locus. And so CRE-mediated recombination can remove the stop cassette and allow lower levels of APOBEC3B expression in any tissue. And we generated in parallel another model where we have upstream of the transcription stop a stronger CAG promoter that, when the same CRE-driven event happens, will induce APOBEC3B or allow APOBEC3B to be expressed at much higher levels. And using these inducible models, and this is looking at whole body expression initially, and we're looking over the entire lifespan of these C57 Black 6 animals, the controls usually develop tumors as well over time, and the life expectancy there is somewhere north of 2 years. The low APOBEC3B expressing animals also don't have much discernible difference in overall survival compared to the controls, but the higher APOBEC3B expressing animals clearly do. And they develop a constellation of different tumors with lung, liver, and blood cancers penetrating predominantly, other tumor types as well, but much more rare. And some representative tumors are shown down here, where we've got a lung adenocarcinoma, an almost unrecognizable liver, and a spleen that's about 4 to 6-fold larger than a normal mouse spleen infiltrated by tumor cells. And so what's really important here is not only is APOBEC3A and B driving these tumors, but when one sequences the resulting tumors and compares those to match normal tissue from the same animals, there's a very clear APOBEC mutational signature. And this is in the trinucleotide context, with C to Ts and C to Gs predominating. And the top is the A3A model, and then the bottom is a representative A3B tumor. So as a little transition point here, and I apologize, but I can't see the timer. Let me play with, I won't worry about it. I'll wait until our moderator interjects. I just wanted to mention that, in Informatics, my lab is really a function-driven lab.
10 minutes.
A number of different model systems. And informaticists in my lab are kind of like kids in candy stores. So if any students out there are interested in learning biological approaches in parallel to the informatics studies that they've already learned and tools that they've already developed, reach out to me and we can discuss postdoctoral possibilities. So in the second part of this talk, I want to focus on another one of these systems that's yielding a lot of data. And Dr. Nick Sinnell's done some work in this system too, which is the haploid cell line called HAP1. It's a human cell line that has a near haploid chromosome number. And one can see that haploidy over here when one's looking at PI staining for DNA content. It's half of what a normal diploid cell is, with a 2N dividing population, a replicating population. And we like this model because it kind of cuts down on the sequencing costs, and all mutations, of course, then are heritable mutations. It does have some drawbacks. And we wanted to adapt it for cell studies analogous to those that we and others have performed in E. coli and yeast, where one can take your favorite model organism, introduce a mutagen, in this case APOBEC, but one can, of course, use other endogenous and exogenous sources and study it selectively. And so we introduced the thymidine kinase gene of herpes simplex virus 1, which renders the cells susceptible to the drug ganciclovir, so that we can literally use that drug to select for mutants that have been, and compare mutation frequencies in the absence and presence of a given mutagen, and in this case, in the presence of different APOBEC3s. And so, like the system I just described with FAH-induced tumors in the liver, one can look in an unbiased manner at all of the different APOBECs and whether they impact mutation frequencies and what the resulting mutation spectra and signatures are. And so this system expresses naturally lower levels. These are just mRNA levels relative to TATA binding protein, a housekeeper. So lower levels of the different APOBEC3 family members. The student that did this work, Matt Jarvis, was fortunate in that one of the TK clones that he isolated, called M9 initially, has even lower levels of the different APOBECs, such that the APOBECs 3A and B are not even detectable, shown over here on Western blots. And there's no detectable DNA deaminase activity that's shown below here in activity assays. And so we can introduce the different APOBECs and compare their mutagenic effects. And here's wild type A3A versus catalytic mutant, B versus catalytic mutant, and APOBEC3H, 2 different haplotypes versus a catalytic mutant. And so when one does those experiments, we first want to see, how do the APOBECs affect cells? And we and others have shown that too much APOBEC is a bad thing, and that's also true in a mouse model. But here we engineered vectors to express tumor-like levels of these different APOBECs. So APOBEC3A expression is mutagenic, as judged by gamma H2AX foci appearance. Also alkaline comets in comparison to controls, which have very little DNA breakage and small comet moments. And APOBEC3B similarly, large comet moments and clear gamma H2AX foci. That's quantified, and I should point out that none of the other conditions yielded foci or comets significantly above the normal endogenous levels in the system. And these foci are quantified here, where again, there's significant levels of increase in gamma H2AX foci with A3A and A3B, but not any of the controls or APOBEC3H shown in this particular series of experiments. And similarly for comet moment, there's some variability here with hydrogen peroxide used as a positive control, but one can clearly see that these 2 APOBECs are above all of the other conditions in terms of inducing single-stranded DNA breaks detected by alkaline comet assays. And so what about mutagenesis? And so I have to say this is my favorite part of this study, because it really enables us to look at mutation in a much faster way, like we do in E. coli and yeast, where one can plate the cells selectively and look at the mutation frequencies per however many cells plated. And so here we're looking, it's roughly 2 to 3 per 10 to the 5 times 10 to the 6 cells, 2 to 3 mutants per 5 times 10 to the 6 cells. And one can see that there's a pretty low level of spontaneous mutagenesis in this system in culture. And APOBEC3A and APOBEC3B, but not their catalytic mutant derivatives, result in increased mutagenesis. And the other really cool thing about this is because TK is a single open reading frame, one can amplify it by PCR and then by PCR product sequencing, get a pretty quick readout as to the actual mutation pattern that's accumulating in these cells. And one can see here readily by plotting essentially the single base substitution mutations. If they're APOBEC context, C to T or C to Gs, they're in red ticks. All other single base substitutions are in black, and indels are in blue. And one can see in the controls, there's lots of black and blue. But in the APOBEC3A and B expressing conditions, there's a lot more red ticks, so a lot more APOBEC signature mutations. And moreover, a lot of those are accumulating, their hotspots are beginning to emerge within the TK or preferred sites for these 2 enzymes. And it's very interesting that it's the same 2 sites that are preferred by these enzymes. So going a little further, so this is just a simple logo plot of those APOBEC mutations in the TK gene at these 2 different hotspots shown below, and all of the TC focused mutations for APOBEC3A and APOBEC3B. And my main point here is that they're pretty much indistinguishable logos. One obviously needs larger numbers to say if there is a statistical difference, but at least by eye, there's no statistically different pattern here for A3A and A3B. And the nucleobase at the -2 position and the +2 position is pretty much equal for each of them. But one can, of course, then take these TK mutants where we know we've had APOBEC exposure and perform whole genome sequencing. And one can look more broadly at the APOBEC inflicted lesions. And in this case, one can see, again, a strong APOBEC mutation signature for APOBEC3A, not as strong, but very also obvious for APOBEC3B in comparison to their catalytic controls. And there's some other business happening in these systems too that we haven't yet fully understood. And this is another way of looking at it where we can look at the relative contribution of APOBEC, which is in this plot is called signature A versus other signature. And one can see a strong significant contribution in all of the A3A expressing clones that are mutated at TK, and in 4 out of 5 of the APOBEC3B expressing clones relative to controls. And now we have much larger numbers of mutations, of course, because we're looking genome-wide. And again, looking at mutations, all APOBEC signature mutations that fall within TC motifs, one can see fairly similar representations of A and T or W at the +1. And at the -2, for APOBEC3B, there's no real difference in purine or pyrimidine. But for the -2 for APOBEC3A, there's a strong pyrimidine bias, which I'd like to note is near 75% of all TC mutated sequences have a pyrimidine at the -2. And this strong pyrimidine bias was previously reported or originally reported by Professor Gerdinian's group. And that's very clear for A3A and in contrast, not much of a bias for A3B. And I wanted to make that point because when one looks at tumors and takes the pentanucleotide sequences inflicted by these enzymes in this HAP1 system, here APOBEC3A, it's kind of buried in this tree, and APOBEC3B, there are A-like and B-like clusters. But if you actually drill into these data sets, there are no tumors. This is a breast tumor data set, and we're in the process of doing it for other data sets, but there are very strong APOBEC mutation signatures in many of these tumor types, but none of them get to the level of a -2 pyrimidine preference of 75%. So based on that and clustering of many tumors with APOBEC3B and other hallmark features that I don't have time to go into today, I would postulate that most tumors are actually a composite of APOBEC3A and APOBEC3B promoted mutagenesis. And so in summary, we've shown using animal systems that A and B are capable of driving cancer development in vivo. I used a couple of those model systems, but it actually really took a long time and several lab members to develop some of these in vivo models. I didn't have time, but also monoclonal antibodies are extremely useful in picking out APOBEC3B versus APOBEC3A tumors. And we now have monoclonal reagents for both. The HAP1 TK, what we call TKM9 system, is a powerful system for looking at the effect of a given mutagen. We've used it for APOBECs, but obviously, as Professor Nick Senal's done before and others, this is a great system for looking at the effects of other endogenous and exogenous mutagens. And it is human chromatin. So I think a lot of the things that we can discern from the system can be applied more broadly. B and A both inflict what we call an APOBEC signature. So I think it's really important to ultimately drill down into which APOBECs are causal in vivo, because as we move forward with using, essentially identifying tumors that are expressing and being driven by APOBECs, there may be different ways of thinking about treating these as opposed to, say, HRD and other endogenously driven tumors. And as I just suggested on the last slide, the overall signature is likely to be a composite and not simply A or B. So I think the field is moving toward an area where we'll have we'll probably never settle this debate perfectly, but I think ultimately can appreciate that more than one APOBEC is contributing to the overall mutation program. And finally, I'd like to thank members of my lab. It's color-coded a little bit, so the mouse team is in blue, and those who worked on the HAP1 system are in red. Really great collaborators all over the place, and also for my funders for strong support, and all of you for listening in this capacity today. Thank you very much.
Thank you, Dr. Harris. There are a number of questions. I'll start from the one in the chat. This one's from Dmitry Gordanian. Could you comment on the TCGA observation that in bladder cancers, higher APOBEC mutagenesis is associated with higher survival rate?
So that's actually, so I didn't have time to go into that. So APOBEC can be good and bad. So in a number of different tumor types, like the one I showed, It's associated with poor clinical outcomes. For bladder, also for clear cell ovarian, and some of the other very highly mutated tumors, there may be other reasons for why instead of associating with poor outcomes, they associate with better outcomes. In bladder cancer, I'm not 100% sure, but it's probably related to the therapy. So we showed in clear cell ovarian that higher levels of APOBEC actually make the cells more susceptible to platinum-based therapies. And so the APOBEC positive tumors do better than the APOBEC negative tumors, and that stratifies over the group because almost all were treated with platinum. With bladder, I don't know, but I would predict something similar where APOBEC is sensitizing cells to a particular treatment and better outcomes emerge. I don't think those are immune therapy treated bladder cancers, but I would also predict that some APOBEC positive tumors, and I'd love to hear a case study by Serena or someone else, where an APOBEC tumor may respond very favorably to immune therapy approaches.
Okay, there's a question in the Q&A. Is there a difference in the intra-tumor heterogeneity of tumors induced by APOBEC3A versus 3B, given that mutations accumulate at different rates?
So that's a really great question, and we haven't drilled into that, I would say, well enough yet. And I think, some people in this particular venue are actually much better suited to do that, with different informatics approaches to separate early versus late. But one thing I would like to say is in many different tumors, even though APOBEC3B is a little bit slower in catalyzing the conversion of cytosine to uracil, A3B levels are higher and persist longer, and in many even constitutively over long periods of time. And so like our liver mouse model, it took longer for APOBEC3B driven tumors to emerge. So I suspect, whereas A3A, more active, lower expression in most instances, and one can imagine faster burn, shorter time versus longer burn, longer time. And I don't know who would win over those situations. So we'll probably see that coming out of data as the field moves forward.
Okay, we just have time for one more question in the Q&A. This one from Francisco Martinez. Do you observe APOBEC causing other types of alterations, such as small indels and double base substitutions?
So that is also good. We don't actually see, we're still in the process of analyzing that for the actual mouse tumors. There's a ton of data there actually when we're comparing them like human tumors of tumor versus normal. In the HAP1 system, that actually might be one of the reasons that it's a model system. So we see a lot of single base substitution, and it's possible that larger events, including indels, are selected against in that system because there's only one copy of most genes. So we recognize that we're probably losing some essential things, but overall, we're still getting a pretty clear picture of APOBEC.
That's wonderful. That concludes the speaker presentations. Thank you, Dr. Harris.
Thank you.
And now we're going to pass the baton to my NIH colleague, Dr. Dmitry Gordenin, who is the principal investigator in the Genome Integrity and Structural Biology Lab of the National Institute of Environmental Health Sciences. Dmitry, I'm going to pass it to you now.
Oh, can you hear me well?
Yep.
Yes, thank you. So I'll share my screen to start from some, sorry, I should be sharing the entire screen and then show the introductory slide, and we shall start. Just need to click on presentation mode. So
yep, perfect.
For this panel, which I feel is by its title is the central to this conference, because signatures are really meant to represent exogenous and endogenous mutational processes. So I decided to put some structured list of questions that I think are indicating interests of community, gaps, and future. And 3 sections is resolution and limitations of signature analysis, input data, what are the sources and availability, and what we consider future. So any of participants is welcome to start, and I may be chiming in or just letting you, and of course, any of session speakers can see what other things you want to address here. It's just what I think is could be of interest to multi-hundred audience that is listening to us. So Ruben, Serena, I see your faces. Or I can keep going through this and giving my view, which I do have also.
So there's a couple of questions in the chat. Are we taking questions from chat or just chatting?
Based on what panel discussions were yesterday, it's a discussion between speakers.
So I would love Serena's opinion on HAP1 versus ES cells, because I think ES cells are also very, very powerful, obviously, and representative of things that people are using for therapy. So, as you implied, messing around with mutation in those, or, mutations that happen in culture can be potentially detrimental to the people ultimately receiving the ES cell therapy.
Yeah, so it's a great question, Ruben. So, and I think it might actually help with point number 2 here. In order to study mutations, we need mutations. So there's this balance between working with a model system, which is not too abnormal. So I think many of the cancer cell lines, if you did mutagenesis work in cancer cell line models, many of those, have got DNA repair pathway abnormalities already, or have also weird physiological abnormalities. And we might see an increase in the number of mutations, but they may not actually represent what the signatures would really look like in most cells. And to some extent, HAP1 was where we started when we did the proof of principle, but that was simply because in those days, whole genome sequencing was really expensive. HAP1 was haploid. We wouldn't have to sequence as much. And you only needed to knock out 1 allele. So that was a good place to start. HAP1 clearly is quite permissive for mutagenesis, so we could generate rearrangement patterns, for example, from it. When we went to iPS cells, they were a little bit more precious. They needed love and attention. They would not generate rearrangement signatures. I think iPS cells, ES cells, they die if they get a double strand break. I guess they're stem cell model systems. You just don't tolerate them. So on the one hand, it's a cleaner system, the ES and iPS cells, but on the other hand, they tolerate certain kinds of damage very badly, and so some patterns don't come through. So HAP1's got its strengths, but it's got its weaknesses, and likewise with the ES and iPS.
Yeah, I think, those are great points, and ultimately, no cellular system's perfect, but I think a lot of things can be modeled in cellular systems, especially when it comes to looking at evolution and resistance mechanisms and impacts of repair and mechanism, and then ultimately, they can go in vivo. We have some issues with mice too.
Yes, yeah. I mean, that DT40 paper in 2016, as early as 2016, David Schutz, I'm so sorry if I pronounced your surname incorrectly there, did that piece of work on DT40, and really beautiful signatures of chemotherapeutic agents. But cisplatin in DT40 is very different to cisplatin in human cells. So and I think all these signatures, they're real, they're relevant. They don't mean that one cohort is better than another. I just think they're different, and I think we need to view it with that lens. That actually there is just a little bit of variation on the different genetic backgrounds, as I think Professor Pommier mentioned earlier. If you have a different genetic background or different species, then you might see a slightly different signature when you do these same experiments.
Absolutely.
Yeah, I had one question actually, following up on that. With signatures SBS 1 and 5 that are up here on the screen, they're both called clock signatures, but I think some of the original papers showed that they don't behave in the same way. There are tissue-specific differences, which I think you mentioned, Serena. And I mean, clearly in the mouse, but we've done very few experiments on this, but in the mouse, we do see SBS 1 in old mice. We don't see SBS 5 in old mice. Also, we see that SBS 5 is proportional, the number of mutations is proportional to the extent of DNA damage induced by a single treatment with a carcinogen. You look at the number of carcinogen-induced mutations from one treatment, and it's directly, I think I showed a slide on that, and it's kind of puzzling, not being an expert in signatures. So where do these signatures come from? What's the underlying mechanism that leads to SBS 1 and SBS 5? And how can we understand their roles in carcinogenesis?
I will be happy to comment on the mechanistic angle. So SBS1 is, so in CPG sequences and only in CPG sequences, cytosines are now and then methylated. And this methylated cytosine, if it is deaminated, it becomes a thymine. So you have a mismatch. And if this thymine is copied, you have C2T mutation. But one speculation that I had for myself about the origin of SBS5, and it is in agreement with some mechanistic studies that in fact this TG mismatch that is created by deamination is also a subject for mismatch repair. And these gaps are known to be filled by error-prone polymerases. And this could lead to a broad, rather flat signature, which would be signature 5. This is not easy testable thing because it would depend on how much is competition between methylation with mismatch repair versus copying, but it's just one possibility on how signatures that are very defined, like Ruben called Appleback signature, pure signatures can intertwine through mechanistic angles with rather flat signatures. And all this funnels to the simple thought that we have just 4 nucleotides and 16 possible neighborhoods of these 4 nucleotides. So it's we are dealing with a finite world, and which is much less finite than the intertwined multi-protein DNA repair handling systems. So different mechanisms can create the same signature. So I wouldn't go to this SBS5 as a single explanation, but mechanistic explanations are there. And in fact, if somebody is interested and whoever is listening us may follow up.
So Gaddy had a really interesting paper many years ago now, actually, I think, where he showed that mutations in ERCC2, I think, could result in a really enhanced version of signature 5, like a hypermutative phenotype. And so clearly ERCC2 is important at trying to limit the amount of the signature 5. It is a bit flat, but there are some of these, the particular peaks is the T2C, the early T2C peaks. So clearly ERCC2 has a role there, and actually, but that principle, so the C2T as CPGs, as Dimitri's beautifully explained there, this deamination of methyl cytosine, we also see it in mutations in MBD4, where this signature happens in a big way, right? There's a massive hypermutative phenotype associated. So, you know, I think there's still so much more to learn about what causes these, what can amplify the signal. Yeah, so much left to learn.
Serena, This is Gary. I just put the link in the to the paper in the chat, and to my mind, SPS5 could be a combination of multiple effects, but part of it could be just slippage of nucleotide excision repair. So spaces that are failed to be repaired, and then when you have mutations in ERCC2, you have more of that failure of repair, and therefore you have higher levels of SPS5.
I got the right gene then. I wasn't sure I remembered that correctly. Very good.
There was one publication I saw that linked SPS5 to inactivation of fragile sites.
And also smoking, by the way.
Yeah. So is that a generally accepted mechanism that controls SPS5 or leads to SPS5 related mutations? So fragility in the genome, if there's a shattering due to radiation or due to some carcinogen DNA damage, is that go through fragile sites leading to SPS5?
Genome fragility may lead to increase in single-strand DNA, transient single-strand DNA that is then repaired somehow, but at the time it's single-stranded. All lesions will be, or majority of lesions will be copied as mutations, because single-strand DNA there is very limited number of direct reversal repair mechanisms. Most of repair mechanisms require second strand. So we can, and since signature 5 is so flat, it can be everywhere, and it's also associated with smoking. And this is why I put this classic pair to address signal versus noise issue. But I think we really came to some consensus that noise does exist in signatures. If I could point to the second section, and in my practice, I entered accidentally in the world of mutational signatures and cancer from yeast genetics. I found really big impediment to research that we do not have enough mutations, although these mutation calls were obtained by people and they published papers. My question to panelists would be, do you encounter similar problems? And also there is one small general problem in the point D, that when you do some analysis of somebody else's controlled access mutation calls, you cannot reintroduce the annotated mutation calls into a controlled access data set. It can never be shared. It is, at least in United States, which NCI is the part of United States so far. This represents the problem for follow-up, for reviewing, and just any input from speakers, or maybe some panelists can chime in about the access to already generated mutation calls, how it's easy, and if something should be done.
I suppose I can have a first stab, and then others, please do join in. I think for the longest time, the focus was just trying to get people to share their data, but I think we're getting there. I think a lot of people are sharing things. Certainly, we would like people to use and the data that we generate, and I agree, Dmitry, sometimes you kind of analyze something, you might have new things to share. In general, I certainly haven't had the experience of recalling mutations and wanting to share them again. If at all possible, I would like to never have to recall somebody else's data set. What a pain. And I would just use their data and credit them. But if there's differences in processing or processed results, then I guess they go into papers, do they? Or even websites, as we've heard in the last couple of days, to sort of deposit processed information. I'm not sure I have any other solutions to offer, but maybe NCI will have a better plan.
There is a request for information from NIH that went out on November 30, just a couple days ago. Request for information for next generation of genome data sharing regulations. And in fact, I will place the link into chat box and hope it will be picked by panelists and by other listeners. It can be now influenced by a mutation community, by mutation signature community, because these mutation calls are not addressed in genome data sharing of NIH so far.
Dimitri, this is Ludmila. I just want to add that this is very country-specific problem. I mean, you put Hartwig as an example. The German data sources are another example. A number of countries in, like China, etc., they have very different regulations of what can be shared, what are privacy concerns, what can be provided outside of the country, etc. So this is, and TCGA has been wonderful. If you have access, you can get all the mutations. ICGC has been even sharing more than that. But for some of those data sets, it's just not possible, legally possible to share the data. And I don't think this is a scientific problem. This is more of a governmental problem, regulation problem.
Yes, in international area, it is governmental problem. And I'm the example of the person who could not get mutation calls from Hartwig Foundation because of this differences in regulations between United States and European Union. We can get luckily, we can get things from United Kingdom, and thanks to Serena and to Sanger, who is sharing lots of mutation calls, and we can get this from Mutagraphs. But for NCI, which is one of the drivers of mutational signature analysis, at least at the United States level, things may be made a little bit more organized and better access to a mechanistic community. And one very simple example why this, even if mutation calls from controlled access can be obtained, why sharing them, back sharing is important. Because let's say I was a participant in PCOG. I get PCOG mutation calls. I ran APOBEC signatures for PCOG mutation calls, and it was placed in association with PCOG marker paper. Now, if I will rerun it with a different algorithm, I cannot put them anywhere because these are from controlled access study, TCGA and ICGC. So that is the problem in point D, and I started hitting it from 2012 from our first paper on mutational signatures. And I don't know, Gadi, if you're listening, you may, I remember that you are also commenting on that.
Yes, I think there are solutions that for this problem are getting towards these cloud platforms that enable to perform computation in the cloud, where you have these controlled access environments that in order to get into the environment or the workspace, you need to have dbGaP credentials. But once you do that, you can post data there that anyone else that only has credentials, for example, TCGA credentials, could go into the workspace. So for example, for TCGA data specifically, you could do that by creating a workspace in the system called Terra, and then you create that workspace under what's called the authorization domain of TCGA, and then only people that have dbGaP access to TCGA could get into that workspace. You will need to pay Google for the storage of those files. I assume they're not huge files, that these annotations of AppleBake on the mutations. But Terra is not the only system like this. But I think this is how we should as a community solve this problem by enabling these environments that are approved by the government to control access of people to according to whoever has permission to use a particular data set. That's my view.
I have to say also that, as I showed yesterday, we are building such a sort of a portal or repository. I showed you the current modules that are there, and the mutational calling will be there. The point is, as Gary was mentioning, we needed to have the government to controls, and we need a funding for keeping the repository, for maintaining the repository. So it's a bigger problem, but we are going there. We definitely recognize the need.
I would like to point also to the line C in that second section, that many papers, they use mutation calls, but they do not deposit mutation calls into dbGaP, but only deposit reads, although they claim that everything is there. And again, this should be somehow specified in this new generation of genome data sharing by NIH.
I totally agree with that. I mean, we need to make sure that sharing is much easier, and people that have the right credentials could get access to large amounts of data. It just doesn't make sense that people need to download the raw reads and recall mutations. It's a lot of wasted effort.
Now, I have a question to Terry. Since you organized this wonderful conference, if I sent you the link to this request for information for new generation of genome data sharing, would you be interested to collect suggestions from panelists and maybe submit? There is a way to submit from the group, and there will be much more attention to submission from this group of mutational signatures than from individuals.
Thank you, Dimitri. I think we have to wrap up, everyone. We're due for a break, and I'd like to thank all the session speakers for excellent talks and discussion.